Hi guys, my name is Stephanie. For this presentation I'll be talking about hearing loss, which I guess is pretty self-explanatory. Like I said, I'm sure you know your grandparents or some older people who have developed hearing loss in their life. Other people can be born with it. It's really common over the people the age of 60, but about 2 out of every 3,000 children in the US are born with this. This can be caused by many different things, like noise, age-related changes, diseases like viruses can actually harm the inner ear and cause you to develop hearing loss, and hereditary, which is pretty much more the focus of this as, genetics class. I actually work in an ENT office, so ear, nose, and throat. One of the big things we do deal with is hearing loss, which is probably why I chose this. I have a really close connection with this, I think. I work with otologists who do focus more on the ears. So going along with this, there's pretty much 2 main types of hearing loss. Well, 3, technically. First one's going to be sensorineural, which is generally there's some kind of disconnect between the ear and the nervous system, meaning the ear can't properly send impulses when they hear things. This is commonly caused by age, or autoimmune disorders, or malignancy. One thing we do tend to see in the office is something called an acoustic neuroma, which is a tumor that's pretty much on the cranial nerve, the one that's involved in hearing. It presses on it and pretty much causes you to develop the hearing loss, usually in one side of the ear. So if we do see this asymmetric hearing loss, we're going to order some imaging to further assess for some kind of tumor. Another type is conductive hearing loss, which is actually more treatable. It can usually be caused by some kind of obstruction in the ear, in the middle ear, the outer ear, like an infection, cerumen, or earwax, or any other of those. That can usually be treated by, if it's an infection, obviously by antibiotics. Cerumen can just easily be removed in the office. So there's 2 types of causes for hearing loss, not just 2 types of hearing loss. So first one's going to be syndromic, which is linked to other disorders. It's kind of like an associated side effect. Or non-syndromic means it can be caused by an isolated phenotype, which accounts for about 70% of cases. This is going to be caused by a mutation in a gene, usually. Okay. So I am going to focus more on, from now on Usher syndrome, which is one of the more common types of causes for hearing loss. So this is an autosomal recessive syndromic hearing loss. This can result in partial or total hearing loss and vision loss, usually caused by retinitis pigmentosa. It generally tends to progressively worsen throughout your life. There's 3 main types, which have about 12 chromosomal loci involved. These types are usually going to be characterized based on the amount of hearing loss present, if they have imbalance associated with this, and age of onset. So basically it kind of goes from most common to least common in this list right here. So Usher syndrome type 1, they're usually born with severe to profound hearing loss, meaning they have the worst part of it. Usually in childhood, they'll start to begin with vision loss, and chronic imbalance is also associated with this, meaning they can be dizzy, they have issues with coordination, they can't walk well. Usher syndrome type 2 is still pretty common, but less common than Usher syndrome type 1. Their hearing loss is more in the mild to severe range, more in the high frequencies, meaning they have issues hearing louder pitches of tones. This usually starts at birth and progressively worsens as well. They will also have vision loss starting in childhood or adolescence, and there's usually no vestibular effects, meaning they have pretty good balance, they're not really dizzy. Usher syndrome type 3 is the least common. I think it accounts for about maybe 2 to 4% of all Usher syndrome cases. This usually occurs way later in life. It may also have imbalance as well. This is usually a less severe type of hearing loss. Okay, so here I have what I'm showing is basically a normal range of hearing. As you can see, so on the left side, it'll be the loudness of, so So basically for hearing loss, we're going to give them an audiogram, which assesses the level of hearing and responds to tones. On the left side, you can see we have the loudness, which is usually measured in decibels, and on the top is the pitch, which is measured in hertz. In the 0 to 20 decibels range, you're going to have normal hearing at all pitches, which is obviously what you want. A little between the 20 and 40 decibel range, you're going to have mild hearing loss. The 40 to 60 be moderate, 60 to 70 moderately severe, then severe, and then profound. Everything above like the 2000 hertz range, like the last, let me get a pointer from like here to here, is going to be your more higher pitches. So the more we age, it's pretty common that we're going to lose these higher pitches of hearing. So a lot of people will have issues, difficulty understanding people in conversations. They might have to turn the television louder. They also will generally have more difficulty understanding women because they do have a higher pitched tone. And so these are examples of people with Usher syndrome. On the top, type 1. On the bottom would be type 2. As you can see, the Usher syndrome type 1 people will have a much more profound hearing loss. As you can see, the decibels is pretty much on the very low end. Usher syndrome type 2 will have a little bit lesser severe. They can still be pretty severe. It's usually going to be worse more in the higher frequencies, because that's usually the first level of hearing to go. I do not have Usher syndrome type 3 audiogram available, but that's usually going to be a little bit better than this. Usher syndrome type 2, hearing loss could be that severe, depends on the age. Like I said, it does progressively worsen. Okay. So I'm going to spend most of my time on Usher syndrome type 1, which is the most common version of it. Usually mutations in genes will lead to a loss of hair cells in the inner ear and loss of cones and rods in the retina, which are involved in vision. This does lead to progressive hearing loss, vision loss, and imbalance. As I'm sure most people do know this, but the inner ear is implicated in vestibular function, so dizziness. There are 6 main different genes implicated. The most common one will be the myosin V2A gene, or MY07A, which is located on chromosome 11q13.5. And this gene has been implicated in over 50% of cases of Usher syndrome type 1, making it the most common one. A mutation in this gene can be due to an alteration of a single amino acid, a production of a premature stop signal, or insertions or deletions of DNA in this chromosome. Normal function of the myosin gene in this is to make the myosin 7 protein, which, while working with actin, it does transport molecules within a cell. It also functions as the movement and shape of the cell. They kind of act as like a track to transport molecules throughout the cell. This is pretty commonly made in the inner ear and retina, where it is involved in the development and maintenance of stereocilia in the ear, which moves in response to sound waves and helps in hearing. So if there is a non-functional myosin protein, these stereocilia may not be correctly, they may not move, they may not respond correctly to sounds, which can lead to hearing loss. It's also been located in retinal pigment epithelium in the retina, which provides nutrients to the retina and has been linked to carrying pigment that is involved in normal vision. A lack of myosin in the retina can lead to something called retinitis pigmentosa, which will lead to progressive vision loss associated with Usher syndrome. Like I said before, Usher syndrome is an autosomal recessive disorder. This is the mode of inheritance. To be expressed, a person must inherit one copy of the mutated gene from each parent. So as you can see in this little pedigree right here, I have 2 carriers, both of whom will have one regular gene and one abnormal gene, meaning they each carry one mutated gene. As you can see, there is a 25% chance of a child expressing this disease. They will inherit 2 recessive genes. There's a 50% chance that their child will be a carrier, meaning they will have one normal and one abnormal gene, and then another 25% chance the child will be completely unaffected. This is another pedigree showing a little bit more detail here. As you can see, I have 2 carrier patients right here. Like I said, 1 child right here will carry the disease, and then 2 will be carriers. If someone with the disease, such as Ben over here, marries Pearl, who does not have any abnormal copies of this gene, all of their children will be carriers, but none of them will have the disease. As such, Ben and Pearl's grandchildren may develop the disease, such as Kevin down here. Usher syndrome can be diagnosed pretty early in life. Usually it's going to be based on audiograms that I showed you earlier, based on the level of hearing loss. Audiograms will measure response to sound in different frequencies. Electroretinograms are also used. This is usually to test how the retina responds to light and can be used to diagnose retinitis pigmentosa, which is a big factor in Usher syndrome. Normal vision and retinal exams are also done, like taking a picture of the retina to fully assess the structure and function of it. Vestibular function testing will basically test like your response to how dizzy you are, like your vestibular function. This is also based like in the inner ear as well. One version of this is called electronystagmography. Wow, that is really hard for me to say. Electronystagmography, which basically tests for nystagmus, which is basically they're going to lay you in weird positions. They're going to check your eye, and if it like moves back and forth really fast, that's called nystagmus, and that's usually in people who are dizzy, which usually is caused by an inner ear dysfunction. Genetic testing is also available for Usher syndrome. This is mostly optional, usually optional, but they're going to pretty much go with all those other tests I mentioned beforehand before they go to this. It's basically like a confirmation for Usher syndrome. There's about 9 tests available. A couple of them, one's called the otoscope V9. Another one is expanded carrier sequencing, an Usher syndrome panel, the otogenome test, the retinal dystrophy expanded panel, and Usher syndrome NGS panel. These are generally multi-gene panels that basically test for the presence of implicated known genes that are present in Usher syndrome. Some of these, such as the expanded carrier screening test, analyze multiple genes for multiple different conditions, while others, such as the Usher panel, is more specific and prognostic for Usher and only sequences the 9 known genes, sequences 9 genes known to be implicated in Usher syndrome. Most of these tests will require a blood or saliva sample to fully analyze the genes in the genome. A lot of these are offered at the Molecular Otolaryngology and Renal Research Laboratories, University of Iowa Hospital, and clinics in the United States. Specifically the Usher panel, which is probably one of the more specific tests. It's kind of the gold standard for Usher syndrome. There is no cure for Usher syndrome. A lot of it is basically treatment and management, so early diagnosis is helpful. A lot of times they can be diagnosed pretty early on in life because hearing loss allow people to sort of hear the environment around them. It's not actual hearing, it basically allows people to process sounds in a better way than what they can. As Usher syndrome does cause vision loss, vision support is good, such as glasses or contacts. And speech and physical therapy is often used such as to address balance dysfunction. There is something specific called vestibular therapy, which my office does offer, which is it's basically physical therapy, but it's very focused on balance and all of that.